Chapter 9, What Genes Are. Well, we talk so much about how do you know what controls whether you have blue eyes or green eyes? How do you know what controls whether you have brown hair or blonde? That's in your genes. Of course, your environment has an impact on it as well, but certainly your genes play a very large role, and also to determine your personality, more complex, but nonetheless governed by your genes. But what exactly is a gene? Well, in short, a gene is a stretch of DNA that encodes a single peptide, which could be a dye, it could be a hormone, and that leads to the trait. But what is DNA itself? DNA stands for deoxyribonucleic acid, and it is basically nucleotides strung together. So if you think of our bead on a string analogy, going back to the earlier chapters where we define DNA and lipids and carbs, DNA is essentially a bead on a string, and those beads on the pearl necklace are called nucleotides. And then once the nucleotides assemble, you twist the two strands around each other to create a double helix or a spiral staircase. Each nucleotide is composed of one, a sugar, two, a phosphate group, and then one of four bases, adenine, cytosine, guanine, or thymine. So here we have a sugar, deoxyribose sugar, five carbon sugar, hooked to a phosphate group, and then you add one of the four bases to it, in this case, thymine or T, and that is a nucleotide or a single bead on the pearl necklace. Then we string them together in various combinations, either an A or a G, and that essentially makes up a gene. And you string thousands of them together to make a single gene. And there's a base pairing rule. A pairs with T, G pairs with C. You can see them pairing this way. The C pairs with the G, the A pairs with the T. It has to do with the shape of the base. They only fit together in a certain way. That's why the G's and the C's have a little triangle shape, and the T's and the A's have sort of a circular shape to show you that they only fit together with each other. And then the whole molecule is built of covalent bonds. Remember, we talked about covalent bonds being very strong, but the connection across it is hydrogen bonds, which has a very important reason for that. It has to do with allowing it to unzip so that it can replicate. Complementary base pairing means that there's a rule for the bases to pair. One side of the double helix has one strand of DNA. The other side is called the complement or the complementary strand. Here's some of the earliest evidence that the DNA molecule was a double helix. It comes from electron microscopy. Scientists looked at this early image. You can tell that the DNA was like a spiral staircase, that it was a double helix. In our DNA, we have 3.2 billion base pairs. A base pair is a T and an A, or a G and a C. That's called a base pair. Thousands of them make up one gene, and 3.2 billion of those pairs makes up all of the DNA in our body, or really just in one cell. And then we can take that data once we obtain it and we can compare. Humans are very similar, whereas a human and a pig is very dissimilar. If you look carefully at that diagram, you'll see that. DNA structure. So then what happens is that double helix gets wound around itself and then eventually wound around some proteins that are called histones. And then those get coiled and looped and into other structures called solenoids. Eventually, you get what's called a chromatin fiber. We talked about that earlier on in the semester. Chromatin is a combination of DNA and proteins. These are the proteins, and the DNA wraps itself around them. Chromosome fibers are looped and wrapped to form the shape of a chromosome. So then what we have to do with our DNA is that we have to copy it. So when a cell reproduces in mitosis or meiosis, it has to first copy its DNA and then it can partition it into the two new cells. That copying process is called DNA replication. It's both interesting for our cells, but it's also useful in the forensics lab to identify paternity and crime scene. First step in DNA replication is you have to unwind the DNA. And that's why, as across the middle, you have these hydrogen bonds. They're weak bonds. Remember, we talked at the beginning of the semester about covalent bonds being strong, ionic bonds being strong, but not as strong, and then hydrogen bonds were the weakest. This is why. Because with a weak hydrogen bond, it's like a zipper. You can unzip it or unwind it, and then you can begin to replicate it. So you have a blue color representing the original strand, and the new strands that are forming against the template are a different color to represent that that strand is brand new. And of course, obeying the base pairing rules, which is A with a T and C with a G, you can remember that by saying apple in the tree, car in the garage. That's a helpful way to remember which one goes with which. It puts new DNA in according to that. So some good vocab words to know would be base pairs, um, complementary pairs, and strands. This is the strand that's old, and this is the new strand. Then what you have to do when you unwind is you begin to build. It's kind of like building a road. You have to tear out the old road, and then as you're tearing it out, you're laying in the new road. The same kind of thing happens in DNA. We move the DNA polymerase or the enzyme that copies the new DNA based on the template. So whenever it sees an A, it puts a T. When it sees a G, it puts a C in the new strand. And then it drives into that replication fork or that origin of replication, and the other strand drives out of it because you're always 5' to 3'. The problem is now you have a lagging strand and you have to repair that later on. But that's how DNA replication occurs. Here it drives into the strand as it's opening, and here it drives out, which means as it opens new behind, you have to go back and repair that later. We do this with a primer. So the primer is a little piece of DNA that comes along and it lays down the first stretch of DNA, and then you attach to that all of the new DNA. On the bottom strand, which is also known as the lagging strand, as opposed to the top strand, which is the leading strand, then you have to keep putting new primers in as you open up new DNA. Really, the best way to see this is to watch a video of it happening because it happens live in time. Step three, when construction is complete, there's two new strands and they're identical. The top strand is half new and half old. So is the bottom strand. It's half new and half old, but both of those double helices are identical to each other. And that is the process of DNA replication. It occurs before cell division. Your DNA is copied. It is what's called semi-conservative. That means it's half new and half old. Each strand serves as the template for the new strand. Now, if that process goes wrong, you get a mutation. As you put an A in, if you were supposed to put a G and you put the A instead, that is a mutation. A point mutation is a change in one nucleotide to a single base pair, whereas a chromosomal abnormality is a large change to the whole chromosome, either an addition or a deletion. DNA polymerase is the name of the enzyme that adds the new DNA as you go through it, but it can make mistakes. However, the cell can proofread, but even in that editing process of proofreading, it can still make mistakes. So even though you proofread and use a spell checker, mistakes can still happen. I'm always amazed because we have spell checkers, but sometimes words are spelled the same but said differently, and so you're still going to get a mistake. So there are repair enzymes, and they do fix repairs and damage DNA, but every now and then one slips through, and if it does, that's called a mutation. It's about one mistake in every billion nucleotides. So here's a good example of a mistake. It's called sickle cell anemia. It's a disease. It's a type of anemia. This is not all anemias. It's just the sickle cell version. One base pair is changed through a mutation and not repaired, and that results in one amino acid being switched, and that's enough to mutate the cell into a sickle shape, which will clog the arteries and cause anemia because you can't get blood through. You can have a substitution. You can also have an insertion where you add or delete a base, and that creates a frame shift mutation. So from that point on, the rest of the DNA sequence is unreadable. Now, a helpful tool in the world of DNA research is called PCR, a polymerase chain reaction. When you get a rapid COVID test, that's an antibody-based test. It's very quick. They can give you the result in the clinic. It's not very accurate, but a more accurate test is when they say, we're going to send it to the lab. It's going to take a few days. They're doing a PCR test. So what a PCR test is where you literally take some DNA and you put it in a test tube with just a couple of other ingredients, a primer, a polymerase, and some buffer with magnesium in it, and you just heat and cool about 30 cycles, and you make millions of copies of DNA. So you isolate the DNA from the skin cell or the blood, and you're going to put it into a test tube. You heat it, you add the primer, and then it cools. And as it cools, it sits there for about a minute or 30 seconds. And then you elongate according to where that primer tells you which gene you're looking for. And then you repeat that cycle over and over again. And if you were in the lab doing it low tech, you'd just be moving it from water bath to water bath. But there's actually a sophisticated machine called a thermal cycler that heats and cools. It actually takes longer because you have to wait for it to heat and cool. So you could do it in your kitchen between two different water baths very quickly. Probably could get it done in like an hour or less. And then you have millions of copies of that gene that you're trying to study. It could be the COVID protein to say that you have COVID. It could be another gene that you're interested in detecting. We used to say when you're trying to find a gene, it's like trying to find a needle in a haystack. But with PCR, you make a haystack of needles. You make millions of copies of it, and then you can detect it much easier. And it can be used for all sorts of things like paternity testing, disease testing, or in a crime scene to see who the victims and suspects are. Another helpful tool in the DNA laboratory and in the world of DNA is called CRISPR, and that's a genome editing tool. And you basically take the gene that you're interested in editing and you feed that information into the test tube in the form of a little piece of RNA, and then it creates a new version of that gene. It's basically like a molecular scalpel. CRISPR stands for Clustered Regulatory Interspace Short Palindromic Repeats. It's part of the natural defense system of bacteria, and other bacteria use it to kill and mutate other foreign invaders. But by hijacking that, using the CRISPR-Cas9 system, you can edit the gene of your choice. So you give it the RNA that allows it to attach to those parts of the gene that you're interested in. The Cas9 protein does the cutting, and what you end up doing is taking that gene that you want to edit and you modify it. So here the gene is shortened and the piece that you don't want is cut out. And now the gene has been mutated in a way that we chose. And it's very powerful because now you can turn a gene on, turn a gene off, make it work better, make it work differently. And so this slide, our last slide, just shows you the powerful rise. They're calling it the meteoric rise of CRISPR in the world of technology. And so when it was discovered in 87, you only had a few papers that were published on it. But in the last couple of years, you have 4, 5, 6, 7, 8, 10,000 studies that you can read about that are using CRISPR, including in 2020, a very successful gene therapy on cancer patients. So CRISPR, you'll be hearing a lot about it. And it basically is a gene editing tool that you can do in the laboratory. Our college has a gene therapy kit that we developed with the Gene Therapy Institute at Christiana Care, and students can use that in the lab to do a CRISPR experiment. And that is chapter 9, what genes are.